The discovery of the atomic structure of DNA, the double helix, not only provided structural basis for DNA replication, it has totally revolutionized modern biology. The understanding of how DNA works has led to biotechnology and new fields such as bioinformatics. Applications of these disciplines have led to the development of new therapeutics and diagnostics in medicine and important applications in agriculture, food technologies, and more. It's no surprise that Watson Crick, along with Wilkins, received the Nobel Prize in Medicine in 1962 for the discovery of the structure of the DNA double helix. And what about Rosalind Franklin, you may ask? Her contributions were absolutely pivotal and key to cracking the problem. Sadly, however, she died in 1958 of ovarian cancer, probably due to the excessive radiation of X-rays she used in her work. In view of the fact that the Nobel Prize is not awarded posthumously, Rosalind Franklin remains the ultimate unsung hero of molecular biology. Now let's go back and take a closer look at nucleotide base pairing. As you can see, the complementary bases are situated opposing DNA strands. A forms 2 hydrogen bonds with T. In the case of the GC base pair, 3 hydrogen bonds are formed. Now, here is a more simplified representation of these base pairs. Replication of DNA is performed by a special enzyme called DNA polymerase, which is located in the nucleus of the cell. When DNA replicates, DNA polymerase reads one strand as a template, producing a new complementary strand of DNA base by base, each time adding the nucleotide that complements, through base pairing, the corresponding base of the template. Thus, for example, when G appears on the template strand, a C is added to the new strand being produced, thus being extended by a single residue forming a sugar to phosphate bond while removing a molecule of water as expected of the condensation reaction. Base after base, the growing strand is extended, growing in one direction only, from the 5' end always to the 3' end, while the phosphate group of the incoming monomer links to the free hydroxyl of the 3' carbon. This is a process of semi-conservative replication. The original DNA molecule to be replicated provides 2 original single strands to serve as templates. 2 new strands are synthesized to complement the old ones. Each double-stranded DNA being produced is thus made of an original strand, complemented by a newly synthesized one. Thus, for each round of replication, new strands are added to the original templates, thus ensuring correctness of replication, always complementing base for base a previously existing template. Clearly, semi-conservative replication of DNA assures that the information in our genes is preserved. Moreover, our DNA is packaged with protective proteins to form chromosomes. The chromosomes are then stored in the membrane-bound nucleus. These structures and measures all go to protect our DNA and safeguard our genetic information, the blueprints required for the production of proteins. However, as mentioned previously, the synthesis of proteins actually takes place on ribosomes in the cytoplasm. Hence, one must ask, how does the information stored in nuclear DNA get to the cytoplasm to guide the production of proteins on ribosomes? This is achieved by making disposable working copies of the genes required for the production of a specific protein when needed. The disposable gene copies are a second type of nucleic acid polymer called messenger RNA or mRNA. mRNA, like DNA, is made of nucleotides. However, the sugar in the backbone is ribose. Ribose is similar to deoxyribose, but has an additional free hydroxyl group at carbon 2. Furthermore, RNA uses uracil instead of thymine to base pair with adenine. Finally, messenger RNA is single-stranded. Messenger RNA is produced in the nucleus, much in the same way DNA is replicated. The enzyme RNA polymerase transcribes RNA base for base through base pairing with a complementary strand of DNA. Once produced, the messenger RNA exits the nucleus and is recognized by ribosomes. The ribosomes can translate the RNA message and produce corresponding protein. Thus, information flows from DNA in the nucleus via working disposable copy molecule, messenger RNA, that drives the synthesis of proteins in the cytoplasm. This flow of information from DNA to RNA to protein constitutes the central dogma proposed by Francis Crick. I've got an appointment with Professor David Baltimore. He'll explain.